Good morning, everybody. We're presenting on a project that me and Ethan did about training single cells with microfluidic stress. Nice, thank you. Okay, so the single cell that we were working with is called Spirostomum ambiguum, and it is a unicellular organism that's also a ciliate, and they're ubiquitous in freshwater habitats. Yeah. Here's a culture of the organisms that I grow in the lab, and it's a very dense culture of them. But they're ideal for this study because when either mechanically or electrically stimulated, these organisms undergo an ultra-fast contraction within 5 milliseconds. So they're usually in their elongated state right here, and then after stimulated, they contract. And the learning or training that happens in these organisms is called habituation. And this is a very simple form of learning that occurs when an organism learns not to respond to an inconsequential stimulus. A similar analogy would be if you walk into a room and there's a really loud noise, and at first it's very stimulating, and then you just kind of tone it out. Obviously, it's a little more complicated in us because we're neural organisms and a lot of more complex interactions are going on, but it's a lot more simpler in terms of the scale within these organisms. Oh yeah, and also this device here is this apparatus right there is like how many other studies train these organisms. They just cause vibrational wave in the arena that they're in, which then causes them to contract in the whatever arena they are in.
So now we're going to want to try to explore of how else can we stress these cells. So in my lab, we're a microfluidics lab, and we stress cells out using these microfluidics channels. And the reason why, so microfluidics is on the scale of the micron. That's typically, you want to use microliters. It is a very small small amount of fluid that produces a very laminar flow. And it's also very predictable. So the current applications, we'll go through them real quick. It's lab on a chip. That's just pretty much biology on the single cell level. And then you can also produce nanoparticles. Especially, we use hydrogels because we can simulate cells through these small little particles. But then it's also improving efficiency in food science and fuel production. And the best part is single cell manipulation, where we can actually target specific cells to do either DNA study or gene modification. And so in my lab, we do fabrication. So actually, I'll walk you through. The desired for microfluidics is actually photolithography, which is a very similar method that's used in CMOS. And the reason why is it creates a very high detailed mold, like a mold, that we can cast PDMS into. But the problem is it's very expensive and challenging processes that require the clean room. So we did not have time for this process, for this project, but this would be the end goal, is to get this. What we ended up using was 3D printing. We used SLA to produce a quick prototype that we can produce in like 5 minutes, but it's a relatively low quality. We'll show that in a later slide. And we tried to use a different 3D printing method called SLS, laser sintering, but the person running it never got back to us. And then to make the devices, we use something called PDMS casting, which can produce a high detail mold and actually bonds super easily to glass using hydrophilic ends, using only the plasma cleaners. So no glues involved. We produce hydrophilic ends, and when they connect, they produce water that produces a permanent bond. So we're going to just go through this really quick. We believe that there's a Poiseuille flow going through these devices. We targeted flow between 2 plates, but we also, Cedric's going to talk about it. We took away, I originally did calculations taking away time dependency, but Cedric's going to show that actually the cells respond better to changing the flow, and that kind of removes all the equations I did and makes these very much more complicated, especially because the cells take up, is it about 70% of the channel thickness would be the end result, and that drastically changes the shear stress profiles. So
Okay, so here's the experimental method that we went through when we finally found our device that we're going to use for experiments. The first step to this is you have to prepare the cells for an actual stress experiment, and that requires cleaning up the sample from they literally grow in pond water, so it's very dirty. And in a microfluidic case, you don't want any clogs going on, so you have to sediment the organisms and clean them through a series of steps. And then finally, once it's clean, you put them in this microfluidic chip here. This is just a channel. It's just it's literally just a channel with with very constant dimensions. And you let them sit in there for 30 minutes to acclimate to their new environment, so that once you do start applying the stresses, they will respond as if it's the first time they've felt the stress. And this gives you accurate readings of their learning behavior. Here is the stimuli regime. So first they're steady, and then I turn on the flow with the syringe pump here, and then they'll go in one direction for about 2 seconds, and then I will reverse the direction, and they'll be steady for a little bit again, and then they'll go in the other direction. And I'll just do this over and over again. I did that for about 25 minutes. But as Ethan pointed out, we were troubleshooting, and I would ideally that it would be great if during the uniform flow, they would respond with the contraction. But I found there was a very low percentage of them responding, and they mainly contracted at the end, when I stopped the flow, which makes sense because I think the velocity gradients are at that point there's a lot higher change in the velocity gradients than when in the uniform direction, and I think that would cause a higher shear. But these are all just our hypotheses right now. Here you go. So I go through. I found an aperture size for sieve based on the diameter of
just based on their size.
Yeah.
Okay.
So now I'm going to go back to talk about the different channel types. So this is our initial prototype that we made ourselves using SLA printing. As you can see, the channel is very rough, and there's actually, I should use the laser pointer. There's a line here that actually was developed due to a layer shift because we were shoved in with a whole bunch of other models, and that caused, what we really want is a nice uniform rectangular channel through the middle part here. But because of the style of SLA printing, it resulted in kind of like a chevron shape all the way down the channel. So that's what we'd want to eliminate. And then, so actually, this is the device I currently do research on. Here, if you just hit next, I think it starts. Yes. This is the device I do research on right now. It's called the low energy electron irradiation. And it's designed to keep a thin fluid channel to expose cells to low energy electron irradiation. But the reason why we used it here is it has very nice corners, but a very high shear stress regime. But it's very unpredictable, especially because there's a lot of supports in these channels to maintain a 100 micron thickness that these organisms have to deal with these pillars. And around these pillars, it's very unpredictable of what shear stresses would be, especially on the start and stop of the pump.
So based on these problems, we then decided just to use a commercially available EBD channel. And I just had to go through we went through the troubleshooting process of getting rid of bubbles. Just this is my first time working with microfluidics, so it was just kind of complicated in terms of thinking about how to keep the channel completely clear and the bubbles out of it. But earlier in the beginning of the video, you could see that they respond very well to if you can see the contractions, they respond very well at the end usually of the movement. Go to the next slide. And so this was the final experiment design that we decided to do here. And as you can see, they go in one direction for 2 seconds, and I flip the switch every 2 seconds. Ideally, this would be programmable so that I don't have to sit there and click click the device, and that's also eventually a future direction that will go. Another issue about this setup is that since it's not programmable, and since I didn't program it, and I'm not familiar with the syringe pump, I had like the organism contracts in like 5 milliseconds, but it relaxes in like an order of a second. So it kind of like complicated the data set a little bit because sometimes they would be stimulated, but they're not completely relaxed. So it's that's another issue that we're going to have to overcome in the future. But from this data, that considering all those issues, this data it still showed that they over the time of the stimulating over 10 minutes that there was a decrease in population contraction. So they still were able to habituate, at least from what we can tell, to this stimuli regime. As you can see, just as the y-axis here is the percent of organisms contracting, and the x-axis is just the time in seconds. And every 2 seconds, there was like a stimulation, which I counted the percentage of contracting. So our future direction is to design a microfluidic device that can deliver a stimuli regime similar to the vibrational stimuli, so that we can compare like whether they learn better, whether they learn better in the fluidic stimulus or like a vibrational stimulus. We believe that it will be a fluidic stimulus since they are in aquatic habitats and they've evolved to like sense fluidic stresses and forces. Furthermore, we want to get quantitative results, so we will do PIV and CFD analysis on the flows created in these devices to get very accurate results of the stresses that is acting upon these organisms. And additionally, we want to design some pretty cool devices that would deliver uniform shear stress profiles in cross-sectional areas that are perpendicular to the flow. And also a device that will give continuously increasing shear stress profile in the direction of the flow to get some cool insights on the learning process. And this is a cool video that we got of the organisms, but any questions, and thank you for listening.
Audience questions. What is, oh, go ahead.
Yeah. Is there like a, I guess evolutionarily, like what is the reasoning behind the contraction? Does it like help with movement, or is it just something that?
So usually it's like a predator response. When like a predator like tries to come and like penetrate its membrane, they'll contract as like a motion to get away, but also they excrete these things called extrasomes, which are just like vesicles that are filled with like poisonous molecules. And there's a paper that actually went into this interaction, which was pretty cool. And it like they stripped the extrasomes from an organism, and the predator won. But when they had the extrasomes, the predator wasn't able to take them. So it's like an excretion process as well as like a motion to get away.
There was a question here, wasn't there? Oh, go ahead.
I was noticing in the video that some of the contractions weren't as pronounced as others.
Yeah.
Was there a threshold where you said, okay, this is a contraction, whereas this is not?
Yeah, so I had to incorporate that, which was, I believe, because we were on the time scale of the less than the relaxation process. So like you would have varying levels of contraction because of that. What I said is, what I thought is like, okay, I counted at the most contracted state, and I tried to determine from just like experience whether it's in a relaxing state or a completely contracted state. And so I just I counted contracted organisms. So it's not on.
You could probably trace your particles going back and forth, see if you get Poisson flow. Do you get actually more? It looked like there was pretty uniform across the cross section. Poisson flow, you need what's called the startup length, like 30 diameters.
So it'd be in the cross section, not the top that we were watching. Our hope is in the top we'd be very uniform, but in that cross section we'd have a lot more of a, because of the it's much thinner in the vertical direction.
Oh, I see. You'd have to see it from the side.
Yes.
Oh, okay.
That's what we're trying to do in our lab is
Okay.
try to figure out more the side view,
Okay.
because we can see everything from the top fairly. We do transvection, and we can see everything through the top, but we want to know what the cell's reaction is outside.
I'm sorry. We have a student online that wants to present or has a YouTube link. Is anyone their partner? JJ?
Oh, JJ's on. JJ's online too. Where's? Oh.
JJ is online.
Hey, JJ, can you hear me?
I just don't know how to route the sound from the Zoom to the room without joining on the.
Yeah, I think we told them that we're gonna just play it on people watch it after class.
Okay, and
Yeah.
then they don't have to stay to
No,
ask
yeah,
questions or anything?
yeah,
Okay, cool. I
yeah,
just wanted to know.
yeah. I sent them an email about it.
Good question. How do we make that screen and that screen display the exact same thing?
I just
If you go into presenter mode, maybe we'll do it.
I think I can do it from back there, so I can try that.
Do you want them to be at different places on the channel?
Ideally, they're like all going under the
Now
same
it's just so you don't have to like.
uniformly,
Like on this one.
regardless of spatial orientation they are, so that
No, these screens are
we
weird.
get like a more
Possible to
conscious.
I think these are
stimulus.
linked to these, so like you can.
We're trying to combine the two concepts together. So
No, I think it's good to be ambitious.
Okay, well, that's fine.
All right, 13 minutes. All right, Lucas, take it away. 13 minutes.
So what I wanted to do was see if I could simulate some slant path.
We got to use the microphone because YouTube people,
Right.
they can't hear anything.
So what I wanted to do was it's off.
Is it on?
Seems like it. Okay, so what I wanted to do is simulate some slant path laser power in the bucket calculations over complex terrain using a CFD generated atmospherical optical turbulence model. So first we have to delve into a discussion of turbulence a little bit. So turbulence involves fluctuations in fluid that aren't very predictable. A turbulent fluid velocity field conserves mass, momentum, and energy, though, so it wouldn't be correct to say that it's exactly random. A simple definition is that it's a flow state in which there's a fluctuating nonlinear 3D vorticity, which I've shown in the not-so-3D images on the right. In very large-scale geophysical systems, the stratification of the atmosphere and acceleration due to the Coriolis force restrict the vertical motion of the eddies. So turbulence can be modeled as taking place within 2D surfaces at any elevation, and I think chapter 13 of the textbook goes over that. But that's too large scale for what I cared about for this project. So as depicted in the top right image, laminar flow is characterized by the predictable patterns of motion in layers, whereas the turbulent flow has significant mixing and eddying. Some of the characteristics of turbulence are the irregularity. So it's inherently unsteady and unpredictable over time. Even in steady flows, small changes in the conditions can lead to large variations in the flow pattern. It has diffusivity, so turbulent flows enhance the mixing and transport of the momentum, heat, and mass in the system. That's due to the eddies and swirls that transport properties between different layers of the fluid. There's dissipation, so energy in the turbulent flow is transferred from larger to smaller eddies, as we'll see in the next slide, eventually dissipating as heat due to viscous effects at the smallest scales. Turbulent flows have high levels of vorticity, and there's a broad range of scales of motion. So the largest scales are determined primarily by geometry and boundary conditions, whereas the smallest scales are where viscosity dominates and kinetic energy just dissipates. So we'll discuss that a bit more on the next slide. Okay, so to talk about turbulence in more detail, we need to give a quick refresher on the Reynolds number. So it's the dimensionless quantity that measures the ratio between the inertial and viscous forces. So the inertial forces dominate in fast-moving or rapidly changing flows, and they're responsible for the nonlinear behavior of fluids, leading to complex patterns observed in the turbulent flows. Viscous forces, on the other hand, dominate in slow-moving or highly viscous flows and act to dampen the fluid motion and resist changes in flow, leading to these smooth, orderly patterns seen in the laminar flows. So the inertial forces are a manifestation of Newton's first law. So objects in uniform motion remain in motion, uniform motion unless acted upon by an external force. And the second term on the left-hand side of that momentum equation,
Just a cursor.
yeah. Let's see. Yeah, so the second term on the left-hand side of that momentum equation represents the inertial forces and describes the convective acceleration of fluid particles, where u is, of course, the velocity field. Now, for viscous forces, those are caused by internal friction between layers of fluid that are moving at different velocities. And the second term on the right-hand side of this describes the diffusive transport of momentum due to the viscosity. When you take the ratio of those forces, you end up with the Reynolds number that's defined here, where u is the flow speed, rho is the fluid density, L is the characteristic length, so this quantity right here. And mu is the dynamic viscosity of the fluid, nu is the kinematic viscosity. For high Reynolds numbers, inertial forces dominate, and you end up with turbulent flow, such as with the spigot on the right-hand side of this picture. And with low Reynolds numbers, viscous forces dominate, and you end up with laminar flow, such as with the spigot on the left. Because you've completed homework, I believe, 3, or problem number 1 on homework 3, I'm not going to go into the math, but when you non-dimensionalize the Navier-Stokes equations, you end up with the third equation shown there, the momentum equation rewritten to accommodate the Reynolds number. And you can see that the viscous terms vanish as the Reynolds number approaches infinity. So good figures for comparison. For pipe flow, it's often laminar at Reynolds numbers less than 2,000. Between 2,000 and 4,000, you get a transition period. And above 4,000, you typically see very turbulent flows. So now that we know how turbulent flows start, let's start examining them more closely. So we'll break up the scales of turbulence into 3 categories: the integral scales, the largest ones, intermediate scales, and Kolmogorov microscales, the small dissipative ones. So the integral length scale is the largest eddy size in the energy spectrum. They're anisotropic and highly energetic, as you can see above here. So they're influenced primarily by the boundary conditions and geometry of the flow, and they're responsible for the initial production of the turbulent kinetic energy. So these scales are not very affected by viscosity, but through a process known as the energy cascade, you can see as these eddies break up into smaller ones, and you can also see that as we move down the ramp here. So you can see through that process that they transfer energy to the smaller scales. So the integral scales exist in what we term the integral range. The next largest scales are the intermediate scales, or Taylor length scales, and they exist in the inertial range, where energy cascading from the larger to the smaller scales occurs with little energy dissipation. So the dynamics are influenced more by inertia in this range than viscosity, which is why it's termed the inertial range. And the energy spectrum in this subrange follows Kolmogorov's 5/3 law, which describes how energy is distributed among these scales. The smallest scales are termed the Kolmogorov microscale. A little bit confusing, but it's different coefficients. And they exist in what's termed the dissipative range of the energy spectrum. So at these scales, the viscous forces dissipate the kinetic energy of turbulence into heat. So the size of these scales is given by the Kolmogorov length scale, the ing here, determined by the velocity of the fluid, and epsilon, the rate of kinetic energy dissipation. So the chart on the bottom right plots the log of the wave number k, where k is the inverse of the length scale, and the log of the kinetic energy over these 3 ranges. So you can see how the kinetic energy drops off. Let's see if I can get to the next page. So you saw the title. This presentation's on lasers. Why should fluid dynamics matter? So in turbulent flows, there are changes in density throughout the flow, which cause a change in the refractive index, which in turn changes both the direction of propagation, as you can see in the image on the bottom, and phase delay experienced in electromagnetic waves, which you can see in the image on the top. So for camera imaging, the phase information isn't that important. But for coherent laser systems where data regarding target information or modulated communications is phase encoded, this makes it difficult to reconstruct at the receiver, assuming you can even measure the signal at the receiver in the first place. So turbulence isn't causing the energy in the light to get lost. It's stretching that energy along the axis of propagation of the beam so that there's much less energy you'll see at your detector per unit time. So typically you'll need a photon counting detector at long distances to try to even reconstruct the signal if it's gone through significant turbulence. This is why it's expensive to use high energy lasers. Without knowing the times and directions at which you can laze through the smallest amount of turbulence possible, you're eating up a lot of energy, just dissipating it into the atmosphere without getting much energy on target. So using Taylor's hypothesis on frozen turbulence to assume that the turbulent entities don't significantly change shape along the path of the detector, we can describe the relationship using the Gladstone-Dale law, where rho infinity here represents a reference density, and g is a constant that's wavelength dependent. So that's this. Refractive index is a function of density and wavelength. And the refractive index leads to an optical path length and phase difference that causes one point in the cross-section of any propagating wavefront to arrive at a different time than another. Oh, darn. So small scales of turbulence result in higher frequency turbulence, and that affects the fine details of the beam. It's more significant near the transmitter or receiver, not really significant along the propagation path. Large scales result in low frequency turbulence that causes large distortions in the beam. And that's why I chose a large eddy simulation that I simulated through OpenFOAM, which is a CFD software. So I tried to import CAD models that failed. I ended up just modeling a cliff in OpenFOAM and then simulating steady state fluid flow over that. And I've overlaid the velocity field that I've solved for it, a specific frozen time step, onto the cliff image. And I've defined initial conditions here. I then attempted to relate via Gladstone-Dale the density. So the LES simulation solved for density, pressure, and velocity fields.
You're doing compressible.
Compressible, yes. Yeah, so I took the data from the density fields and applied that through Gladstone-Dale to try to figure out the attenuation that a laser beam would face along different propagation paths and at a frozen instance in time. And so I took one along a propagation path that I assumed would have very little changes in density of the fluid. And then I took one where I'd assumed that there would be the largest changes in density. And then I qualitatively compared for a given brightness of the input, spot size, the attenuation that would result at the output. For a fixed distance, I know that the arrows are slightly different in length, but I did that for a fixed distance. So current work in the field, it's worth mentioning that this is all relatively new. Lasers were only first developed in the 60s, and that technology didn't become mature or cost-effective until the early 2000s. Laser comms are typically only used for communication to and between satellites because of turbulence and attenuation-related issues, so the technology has just sort of started kicking off in full force now with a focus on 6G comms. For astronomical systems pointed straight up, statistical models of turbulence based on meteorological data are good enough, but for real-time systems that need to know refractive index variation along a path, we typically use wind Doppler LiDAR to measure wind speed information from Raman backscattered spectral lines and overall backscattered signal strength to find a statistical measure for the refractive index fluctuations along any path. So those systems cost at a minimum hundreds of thousands of dollars, though, just material parts. So not terribly cost effective to put alongside every laser system that you'd ever want to deploy. So what's next here? So there are textbooks with the math on the various statistical methods used to characterize the atmosphere using active sensor systems, but nobody as of yet has related statistical theories surrounding optical turbulence effects back to the Navier-Stokes equations. So all of those experimental statistical models deployed in the active turbulence characterization systems have yet to be validated against an accepted means of obtaining truth data. So there's no good way to say that one sensor system is more accurate than another yet. And this was suggested as a thesis topic to me, so that's what's next, and I've purchased tons of textbooks. Another direction that would be interesting to take this in is using terrain data sets. I initially tried to use the U.S. Geological Survey topographic height map data, but that didn't import well, to provide geometry, and then meteorological data as well to provide initial conditions. so that you can create localized 3D temporal predictions of the freed parameter, a measure of coherence that represents the largest area over which the turbulence effects are negligible, so that you have a decent idea of what performance that you could expect for your laser system and at what time. So those are future directions.
All right.
Okay.
This one you set up while we answer one or two questions.
I'm not so familiar with like turbulent flow regimes, but is it possible to have like a steady state turbulent flow, or is it always, are the eddies always transient in time?
So even with the simulation that I performed, the eddies, as we advance in time steps, they move to the right of that image. So as the eddy sort of reaches a critical size, it moves to the right, and then another eddy is formed off of that edge of the cliff, closer to the cliff, that then reaches another critical size and then moves along in that path. So those eddies don't stay in one location.
And they decrease in size as they go along?
No, they tend to sort of increase, at least in the way that I was seeing that, but the kinetic energy dissipates.
Okay. Thank you.
Okay, we've got 3 presentations left and 35 minutes. So I'll give you 12 minutes each. This is the last class, as far as I know.
Executive decision.
Yeah.
Not sure why it's not presenting. I have it's just
Oh, hit from beginning, up on the top left.
yeah.
Oh, it's still not.
Yeah, it's not presenting on either of the screens. I've never had this happen. I can present it from here.
We'll close it and open it again. It's still in presenter mode in the other.
It's on like the. It's showing the same thing.
like for the other presentation.
Oh, it might be.
Let's close some of that stuff. We're good.
This is.
Oh, it's
Cats.
okay.
Don't know whose this is.
That one's
Yeah.
yours.
Oh yeah. Not too sure.
Let's close it. We can reopen it.
Johnny Cat, I'm gonna have to close yours. I'm sorry.
fine. Task manager.
I think if it's like too many presentations open, it's not happy.
Is it in the downloads?
Do Google too.
Shouldn't.
Can I try now?
It's trying to present. I think I could probably present from here if
Yeah.
I need to.
Where's the cursor? Yeah. Just document recovery.
Just close it. It's just saying that we didn't save it. Yeah, I can just present from here.
I mean, yeah.
Yeah, I can just present from here. It's not gonna change too much.
Alright.
I guess.
Sorry, I'm making it worse. All right,
It's
let's just do
okay.
it.
I'll figure it out. Is this way? No, nope. Okay.
All right.
Oh.
10 minutes, Chris.
Okay. Okay, cool. So I originally said my presentation was going to be on modeling CO2 plumes as well as human breathing, but in reality, the bigger picture is we are looking at mosquitoes and how they actually find you. So by modeling the CO2 plume, that's what they are attracted to. But there's also a lot of other biology behind it. So I'm going to quickly introduce the background and then go into the fluid dynamics part after it, because the biology is interesting, but there's not really fluids. Here you can see there's a lot of mosquitoes flying around me in that picture. Quite unfortunate, but it is what it is. I forget that I have all these other stuff on the slides. But so mosquitoes find you through like 4 different really main ways. Visual cues tend to be pretty important, but they don't really offer too much for fluids to look at. I know we looked at the turbulence affecting it, but they can't really see much anyways in the vision anyways. They just see like blurs. They can see color. Doesn't really make too much of a difference. But what actually is has a lot of fluids behind it is the is the CO2 plume, the VOC, volatile organic compounds that are released by your body, as well as how the body heat kind of impacts all of that. And they all tend to just work together. Like the CO2 can bring them in, but you can't. They don't really feed without the body heat. And then the VOCs, they all just kind of confirm that there's an actual target for a mosquito to feed off of. And they're quite sensitive to changes in small changes in CO2. Like amount of CO2 in the atmosphere at any given moment aggregated over a large volume is pretty small. It's like 0.04%. And the human breath, when you breathe out, is about 4%. So couple orders of magnitude higher than what the atmosphere is, and the mosquitoes can sense a tiny change in it. They use all these small sensors on their antenna to actually sense it, but that's all the biology. And then similarly, they also have sensors to sense sweat gland, or not sweat glands, but the VOCs, the volatile organic compounds that are released, such as ammonia, ketones, lactic acid, all of those things that come with sweating and just day-to-day human body regulation. There's different areas on the body that have more or less sweat glands or other VOC releasing body parts, and they kind of prefer certain areas because of this. Similarly, there are thermal cues that are involved with mosquito behavior, such as just your body heat is releasing from you, like convectively, conductively, etc. And mosquitoes use this as a confirmation to be like, I can find a decent host or a decent place to get a blood meal, survive, and move on. You can kind of see how in previous papers they've been able to get an uptick on landings when the temperature is actually of a human or whatever target temperature's body temperature it is. To actually test all of this, we can actually have humans be a good bait source, and mainly me. I guess mosquitoes like me, unfortunately. But we have an environmental chamber with an infrared camera, dual infrared cameras that can actually track the XYZ position data, mosquitoes in real time, and we can kind of see what parts of the human, or parts of me that the mosquitoes are attracted to. And so this is the data with me standing up. You can kind of see an idea of like all the mosquito tracks. Oh, I should use the mouse. In yellow is talking about the vector of the mosquito, or how oriented it is towards my head, because we are that's where the CO2 is coming from, my mouth, and we kind of wanted to see that. And you can kind of see for the most part they are flying towards the upper head, above my head area, and then you can kind of see the number of mosquitoes at any given moment, relative to the body. Similarly, you can see that they are quite attracted to my head when we're laying down. You can see the tracks. I don't know. It doesn't really show a clear, silhouette of me laying down here, but you can kind of see the number of mosquitoes at any point above my head is quite high. So, all that being said, mosquitoes really like to bite people, and I think we all know that. But going into the fluids part of it is we wanted to look at a model of human breathing to create this plume of CO2 that is released when you're breathing. And so I created a CFD model with a surface boundary condition on the person, surface boundary condition on the person, as well as an initial velocity of air from the mouth with slight turbulent parameters, and kind of wanted to see how this would actually affect the model. And you can kind of see some convective heat transfer causing the air to go up around the person, as well as a flow out from the mouth. This doesn't completely correlate with the if we lay a silhouette of the mosquito tracks with the XY position on top of it, doesn't exactly line up with where the mosquitoes are coming from like we had thought from the CO2 plume. And so we were trying to look at why that is the case. Similarly, we did the same thing with a laying down person, where you can kind of see there's not a jet necessarily, but a dispersion of CO2 or breath from the person laying down. And this also kind of doesn't perfectly correlate with the data that is presented from the mosquitoes tracks. So there's probably something else at play. Before we wanted to look at the thermal convection of the VOC odors, we also I just created two quick fog videos to just confirm that the flow of the human odors creates this triangular cone-shaped plume, which it does. Yeah. So that's a allegedly standing person.
Do you heat the fog?
Yeah, the fog is heated, so it kind of acts similarly to how if you were to breathe, if you go outside on a really cold day, I know as a child, you can like when you're waiting for the school bus and you're breathing out, you can see your breath go up into the air, kind of like a train would, like release steam. Similarly for laying down, I guess it's pretty basic. You can kind of see that the plume that it creates is relatively big, but it doesn't completely align with how the mosquitoes are flying into the person. Originally, yep. I had these hidden, but now we also use temperature gradient to calculate a Rayleigh number to look at the human convection. And this kind of lines up a little better with the mosquitoes going towards the body. So these are what convection, it shows that the convection of the human body releasing temperature to the air is relatively, It's pretty, the buoyant forces dominate over the viscous forces, causing a lot of convection heat transfer to happen and creating these fluid flows. You can kind of see it in the Schleien photography. You can kind of see this boundary layer of air rising due to the convective forces. And that kind of pretty much lines up relatively well with the mosquito tracks and how they're coming towards you. And I created another video with a particle sim to look at how a ammonia particle from a person that is being released would relatively fly off the person or rise off the person, creating this pretty pretty well sought after plume of bait for a mosquito to find you. And I calculated the Stokes number of an ammonia particle in this flow field to pretty much just show that the ammonia particles follow these streamlines pretty tightly, and if there's any minimal flow going up, it's going to also follow that flow going up. In conclusion, here's just all the tracks to visualize because it's quite pretty. But in conclusion, human bait plumes tend to align with mosquito trajectories, whether it's CO2 or VOC, not individually. So they kind of need to be consistently paired together for mosquitoes to actually attract towards you. And not that they won't attract on individual ones, but to get a lot of them or to not get a lot of them, depending on what you want to do, you want to get them together. Human heat convection does create a VOC particle plume, as shown in the ammonia. And then the VOC particles, because of their small Stokes numbers, will closely follow the streamlines. So yeah, any questions? Cedric.
Where do you get your mosquitoes, and how do you release them?
Yeah, so we actually did these tests at the CDC. So they've raised. I don't know if you know, recently in this past semester, there was this whole thing with malaria cases in the U.S. There were 7 breakouts, locally transmitted breakouts. So there were 6 in Florida around Sarasota, 1 in Texas. So they were a huge news thing about there was people going to the insectary at the CDC to kind of look at that because they study all of that stuff. But they also raise, and none of these mosquitoes had any diseases. We made sure of that.
Just let them set it. You can take the mic, but they're just going to set it.
Oh yeah. None of the mosquitoes had any diseases. They're raised by the CDC, and then they just give us the mosquitoes. We release them by putting them in a capsule and just kind of knocking the capsule over, and they just disperse on their own.
Aside from putting yourself in cryogenesis or just stopping breathing, is there anything that you'd recommend to avoid attracting mosquitoes?
Honestly, the more I think about it, mosquitoes are really good at finding you. There's times where there's a small, when we first started these tests, we hadn't fully sealed off the chamber. So in the back corner of the chamber, there's a small hole. Somehow the mosquitoes got out of that hole and then flew all the way across the room to us in the front. I don't know how they did it, but there's not really much that you can do. I guess with all of this, it's human, we don't have any sense. We try to remove as many scents as we can, such as if you shower and all that stuff. So I wouldn't shower for the 24 hours or shower with unscented soap. So obviously pesticides and insect repellent, probably. They've done some research there to help.
So you stimulate your brain out of the mouth?
You didn't put yourself.
Yes. Yeah. I know mouth breathers are people don't really like mouth breathers, but yeah.
I'm not saying that.
I'm going to present for.
It's targeted, it seemed like the area like the torso could be because of noses breathing more,
Really?
Yeah.
breathing out.
Because you're not the Google, so we only
It probably would. It's just
PowerPoint up.
I just used the mouth because it was simpler and it was the largest aperture, so I just assumed people, I don't know, breathe out of their mouth too.
Also, sorry.
Did you have a question?
Yeah, sort of along the repellent conversation. Does repellent just sort of put off scents that like repel mosquitoes, or does it actually like?
I am not sure. I don't think it blocks your scent. I just think it creates a pretty strong, concentrated scent that mosquitoes don't really like.
Okay.
Your model, is it the person wearing like clothes?
No. I was in the pictures. It was like I was basically wearing like a mesh netting. It didn't work as well as I had hoped, but
So like in your model, the heat transfer one. Is
Yes.
the person wearing clothes or is it, like naked?
it's naked for all intents and purposes.
Next slide, Chris. So Daniel Hill's not on the Google Doc. So we got 3 presentations left in 25 minutes. I think we're going to just give everyone 10 minutes, and then Daniel will go over a little bit for yours. Yeah, because we can't get it shorter. Go ahead. 11 minutes.
Okay, we'll speed run it. Okay. Good morning, everyone. My name is Courtney. I'm here with Annika and Jay, and we decided to investigate the progression of fluid flow through a stented artery with an aneurysm. So you may remember this from our journal club a little bit, but an aneurysm is defined as the structural integrity or the loss of the structural integrity in an arterial wall due to the constant remodeling from cyclic loading due to the blood flow going through this artery all the time. And a weakening in this arterial wall can be formed from high blood pressure or a buildup of plaque, which can appear in these spots that become weaker over time. And there are severe consequences to this, such as rupture or hemorrhage. And one solution to this is stenting that you can see here on the right. And this allows for some flow diversion to kind of direct it away from the aneurysm and through the artery in the direction in which it's supposed to go. And the stent itself develops endothelial tissue around it over time, which then again improves that flow diversion over time. So blood has specific properties we had to take into consideration when building our model that we'll be testing, and Annika will get to that in a little bit. So we had to use pulsatile flow, which is driven by pressure gradient, the longitudinal direction of that vessel. And if you think about this, it's just as the heart is constantly pumping, that's pretty much what we're taking into account here. And this can vary in frequency just depending. Tachycardia or like an increased heart rate, for example, would be, one example of this. And blood itself is non-Newtonian, so we also have to take that into account as it is, it changes in viscosity, just depending on the applied shear stress in the vessel as well. And you can see on the right, I broke down the blood structure here, which isn't quite as important for this, but yet these red blood cells here form rouleau structures that we've talked about previously in our journal club. And because these red blood vessels stick together, or red blood cells stick together, they form these rouleaux that then increase the viscosity. And this is important when you're talking about fluid dynamics in an aneurysm. Okay, next, just to get into stent endothelialization a little bit, because this was kind of the main goal of our project. So this endothelial tissue develops around the stent, and this in turn helps prevent vascular thrombosis because endothelial tissue itself is an anti-thrombogenetic material, meaning that it is less likely to cause blood clots as it's natural, as opposed to a stent, which is more of a foreign body. And in general, it usually takes about 4 weeks, but it may vary because there are different types of stents, like drug-eluting stents, different ways to minimize this as well. And the overall goal is to have a fast endothelialization. So this figure on the right here, we actually saw in a study, and this shows 0 days after stent implantation. Figure B shows after 7 days or a week, and then C is after 28 days. So you can see in the bottom one the development of these cells here, and this is the endothelial tissue. Figures D through F here are just at like a 300 times magnitude in comparison to the first one, just to give you a closer look of what this actually looks like. And then this graph here below shows this stent endothelialization percentage over about a month period. And this is kind of what we planned to mimic in our study. So here at day 0, obviously there's no endothelialization. And all the way by day 28, it looks like it's 100% endothelialized. All right, now I'm going to pass to Aniket.
All right, thank you. So as Courtney mentioned, the main goal of what we're trying to do is we are trying to model the impact of stent endothelialization as a function of time on the flow velocity profile. And we plan on wall shear stress as well, but we mostly just looked at the flow velocity profile in an idealized coronary artery model that has an aneurysm. And so these are just some simple diagrams to kind of illustrate what endothelialization looks like. Now, it's very idealized because like these darker red regions, which are supposed to illustrate the endothelialized layers, they don't exactly form, in this type of pattern that you see here. However, we kind of modeled it, the endothelialized layers, as a similar kind of pattern just to be able to modify the percentage of the endothelialized surface area over the surface area of the stent. And so this is kind of just what a partial endothelialized stent would look like, and a fully endothelialized stent would just be identical to like a solid cylinder. You just insert, and ideally there'd be no flow going through the aneurysm at all. So we started with developing some CAD models. We found a paper that modeled an idealized coronary artery with aneurysm. So this is just an average coronary artery with about 4 millimeter diameter, 200 millimeter extension, and a 10 millimeter aneurysm diameter. So this is a pretty large aneurysm. And so it's technically pretty close to rupture. So that's why we decided to model the same design, and you can see our generated model at the bottom. We also obtained a coronary stent online, and so we used that as well. And then we developed these endothelialized layers that, again, as mentioned before, it's in a standardized pattern here. We kind of just created strips of the layers around the stent, and we were able to adjust the width of these strips in order to then adjust the percentage of endothelialization. And at the top, you can just see the full assembly with the flow going through here and then output it there. So then going into CFD, or computational fluid dynamics, a lot of groups have already kind of talked about CFD, but it's basically just a method to 3 minutes. Okay, it's just a method to visualize qualitatively and quantitatively flow through any geometries, and so it requires for a discretization of the geometry. In our case, the fluid domain, or blood going through the geometry. We then use the continuity equation for mass balance, and then we then use Reynolds average Navier-Stokes equations, which decouple the time-dependent and steady-state solutions. And for the time-dependent solution, we wanted to incorporate turbulence as well, which is why we use the shear stress transport transitional, or SST trans model, to incorporate the transition between laminar and turbulent fluid, which is important as you go from the straight vessel into the aneurysm and then back out. You can just see the final fluid domain mesh we have here for one of the models. And for a boundary condition, we actually just decided to look at one mass flow rate, which we got from another paper.
Okay, Okay, I'll have to run this quickly. But so, due to time constraints, we were only able to have two models, one without anything, just the aneurysm model, plus the one with the stent with 40% endothelialization. And as you can see here, without any stent or anything, there's high velocity and wall shear stress within the walls of the aneurysm, which could cause a rupture in the aneurysm, and there's no flow stasis and thus no thrombosis within the sac. And you might be wondering why having this stent, although it's porous, that it might help the reconstruction of the blood vessels, is because that those porous holes in the stent kind of diverse the flow throughout the aneurysm. So instead of having like a high wall shear stress near here, it's going to divert the flow upwards, so it kind of spreads out throughout the aneurysm sac. And with the 40% endothelialization, the holes are going to be smaller, and then eventually there's going to be flow stasis in the aneurysm wall, which are going to basically clot and recreate the vessel walls. And also, although the inlet of the model, the velocity profile was around similar for both of them, the outlet face for the one with the stent, because the flow isn't necessarily going into the aneurysm sac anymore, the velocity was focused in the center of the vessel. And this could be comparable to another study that was done in vivo, actually. Using 4D MRI, they were able to take some velocity profiles. And as you can see, as I explained earlier, without the stent, it's going to hit the aneurysm wall directly, and there's going to be high wall shear stress here. However, if you put the stent in, the flow is going to be diverted and kind of spread out through the sac, which is going to be, and then the stent with the blood cells kind of clotting onto the cell and cause the endothelialized layer, it's going to reduce the flow furthermore, and then the vessel wall reconstruction will happen. And this is kind of giving you like an overview of the current treatment for the aneurysm. So there are some limitations. Obviously, it was an idealized model, and due to time, we couldn't test most of the amazing thoughts that we had. But in future directions, we want to definitely test different endothelialization time points and how endothelialization kind of helps eventually kind of reduce the flow within the aneurysm sac. And also in vitro in our lab, we've been conducting some 3D PTV. I forgot to include it, but it's some laser. You can see the particles through like a patient-specific aneurysm models. And it'll be kind of interesting to see how our CFD kind of validates their in vitro data. It's actually in preparation for a manuscript. And that concludes our presentation. Thank you.
Take one question while Raphael sets up.
So are these stents endothelialized before being inserted, or are they like sequestering cells to endothelialize after?
So the endothelialization basically happens within your body. So as the blood cells kind of hits and kind of accumulates onto the surface of the stent, it starts to create like a layer of basically your vessel wall, similar to your vessel wall. And then the aneurysm part with the flow stasis, the blood inside the aneurysm sac kind of clots, and eventually it's supposed to be like the original blood vessel.
How do blood cells grow on that stent?
On the stent itself, that's a good question.
You're basically relying on that to clog up.
Right.
Once that aneurysm is less pressure, it doesn't shrink?
The aneurysm itself, I don't think it naturally just shrinks.
It's not like a balloon. It's like already
No.
new tissue that grew like that. So what do people just walk around with it?
It doesn't really affect you once it's kind of remodeled at that point, so it's not as dangerous or at risk of rupture.
You just want to reduce the pressure there.
Yeah.
Otherwise, it will rupture, and if it's in your brain, then there's going to be a hemorrhage at your brain, intracranial hemorrhage. Yeah.
You want help?
Yeah, I don't know where to find it.
Again, your stuff's already on there.
Yeah. I'm gonna scroll this. Okay. Thank you.
Do you have videos?
Okay.
Do you have videos?
Yes, I am.
You should probably download it.
Yeah, that's not a bad idea. That's what I was going to suggest.
If you run off the drive, the videos might not be as.
Oh, it downloaded. It doesn't. Instagram.
It would have been good if we had.
That doesn't work.
It doesn't work at all.
That worked.
Okay, so we're gonna do. So if people have to leave at 10:45, they can go to the next class. Daniel will present basically at the end of the class. All right, I'll give you 10 minutes.
Okay, it's working. Okay, so today I will talk about dog lapping and Guinea worm disease. So first, I will present you what is Guinea worm disease and its link with dog lapping. I will present you the objectives of my class project, some fluid mechanics aspects of dog lapping, and then my experience with copepods. So Guinea worm disease is a parasitic infection by a Guinea worm. A person typically becomes infected by drinking water containing copepods infected with Guinea worm larva, and the larva penetrates the digestive tract and escape into the body, where the females and the male mate. Around a year later, the adult female migrates to an exit side. Usually, it will be a lower limb, like the foot, and induces an intensely painful blister on the skin. The wound remains painful during the worm's emergence, disabling the infected person for the 3 to 10 weeks. It takes the worm to emerge the foot. During this time, the open wound can become infected with bacteria and leading to death in around 1% of cases. So public health efforts almost eradicate Guinea worm disease in humans. However, a few years ago, researchers realized that the disease had become prevalent in dogs and could cross back over into human populations. So in order to better understand the spreading of the worms among dogs, many scientists started to study the fluid mechanics behind the dog lapping. So the objectives of my class project was to understand like some aspect of the hydrodynamics of dog lapping, to simulate the dog lapping with the copepods and analyze the behavior of copepods in the presence of the lapping of the dog. So most mammals drink water using the suction mechanism. So humans drink water by lowering the pressure in the mouth, which is possible by sealing the mouth from the atmosphere. However, for mammalian carnivores, their mechanisms for drinking are constrained by the anatomy of their oral apparatus. So with incomplete cheeks, they cannot form a seal to suck fluids in their mouth. And so instead, they use their tongue to lap fluid. So when a dog lap, the tongue first extends in So when the dog laps, the tongue first extends in the curled backward into a ladle shape. Then the curled tongue impacts the liquid surface, including a splash. Then he will retract the tongue into the mouth, and then he will snapshot the water. Informal observations from previous studies have suggested that dogs scoop water with the ventral side of the tongue in the ladle, but recent X-rays imaging has shown that most of the scoop liquid falls off the tongue, and only liquid that stick to the dorsal side of the tongue is transported to the throat. So I use mainly one paper for my experiments, and the first important part was to determine the significance of unsteady versus steady inertia effects on the lapping mechanism. So in order to determine the inertia effects, 19 dogs were filmed when lapping, and they modeled the fluid transport as an open fluid pump driven by pressure difference between the point A and the point B. And so the reduced order model equation here was considered, and the difference of pressure is equal to the unsteady effects and the ratio of steady effects. So with the accelerating tongue, the unsteady inertia effects can scale at this, and by replacing the velocity by a maximum velocity on the time, and the acceleration by the maximum velocity on the time, and the effects of the steady inertia scales at this with a tree to represent the average velocity during the lapping. And so to pump the water, the difference of pressure has to be less than 0. And so the ratio of unsteady to steady effects can scale as this ratio, that is the Stroll number that allows to compare the unsteady effects and steady effects. So what we can see, like with previous studies, the Stroll number is equal to 1.1 for cats, and here the Stroll number was equal to 3.7 for dogs. That means that dogs are lapping with unsteady effects. And so a question was how the dogs optimize the volume of water captured. So the main assumption is that the maximum volume of water happens when at the pinch of time at high acceleration. So the pinch of time is the time when the liquid pinches off from the road and falls back toward the path. So the dog has to try to capture the water when the volume of the colon is maximum. And I will compare 3 pinch of time, the experimental one that was measured by filming the dogs, then 2 theoretical model. So the first model is the unsteady Bernoulli relation for higher acceleration. They model the velocity potential by this, and then they use, thanks to an unsteady Bernoulli relation, they have the velocity, and then they measured with this equation that represents the weighted length on the rounded cylinder that must be equal to 0 at the pinch of time, and they obtain a first pinch of time, a first theoretical pinch of time. For this model is well matched with high acceleration. And the second one is based on hydrostatic pressures and is more valid for lower acceleration. And what we can, so then they do exactly the same thing to obtain the second pitch of time. And what they found is that the 2 models are interesting in different area, but at the end, the dogs, they lap in the blue color band, and so for this reason, the first model is more valid to compute the pinch of time. So to maximize the volume of water lapped, the pitch of time can be estimated with an unsteady Bernoulli model, and with this equation, and the volume can be computed with the radius of the tongue. And so this is the experiment that I did with the Dr. Janet Yen's lab. So they have some copepods that are not infected by worms, obviously. And what we had 2 cameras to be able to film them in 3D. And the goal was at the end to analyze their behavior. So and for this, we simulated the dog lapping with rods, with glass rods, that we put into the water. And the goal is to understand like how the dog lap they will ingest the worms. So we have to determine an area where the copepods are scared by the road. So it's a video, and as you can see, it's very difficult to see them. They are very, very tiny. So this is this, and the goal is to analyze the behavior when the road is in the water. And it's why we have 2 cameras, because sometimes we will not see on the first camera, but we will be able to see them on the second one. So yeah. So to conclude, the fluid mechanics of duct lapping is mainly NCD effects. NCD effects drive the motion of the liquid column for dogs. The main parameters are the size of the tongue and the acceleration of the tongue. And to maximize the volume of water, they have to capture the water at the pinch of time. Obviously, they do that naturally. They do not compute the pinch of time to drink. And the pinch of time can be estimated with an unsteady Bernoulli model. For the second part of the project, the goal was to analyze the copepod behavior by using ROD to simulate the dog lapping, and the goal will be to estimate the area where copepods are scared off. And to improve this project, it can be interesting to build an automatic system that can move the rods with a constant acceleration. Okay.
The copepods all, Daniel, when you set up for -2 minutes now. Copepods all, did they actually, how many copepods did you have in there? I saw them fly away in the beginning.
We don't know exactly how many copepods are there, but it's around like 15, and it's very hard to work with them because they are very scared of everything.
You have to pick any up.
Hmm?
You pick any up with a rod.
No, they will not be attached, and it's why they're trying to understand why the dogs are drinking copepods, because as they don't use suction, it means that it's less probable that the copepods will be drunk.
Do you know like what level the copepods like usually are in the habitat? Are they like the surface of the water, or are they deep in the water? Or, like, what type of water? Is it fresh water, probably?
It's not fresh water. It's water drawn by the people. For example, it's Chad, so it will be water that is stagnant. So I don't know exactly which type of water it is, but I think it's not very fresh water. It's a main problem that the water is not very healthy in this area.
Yeah, I'm going to try and be as quick as possible for everyone, but I just wanted to demonstrate Softman-Taylor instability using a Healy-Shaw cell. So first, what is Softman-Taylor instability? It was discovered in 1958 by Philip Softman and Jeffrey Taylor, hence the name Softman-Taylor. It is... Just make sure this is on. It's a formation of a pattern in unstable interfaces between two fluids, specifically when a less viscous fluid is pushed through a viscous fluid pushes a less viscous fluid through a narrow channel or some type of porous medium. And it's also known as viscous fingering, just based off of, you can kind of see that it sort of fingers out into these like finger shapes. So I'm going to use viscous fingering and Softman-Taylor instability kind of interchangeably. And I wanted to demonstrate really quickly, but if I can't get the cam to work, then I could probably, I have a video of it, so. So yeah, so right now I'm just adding water into the Healy-Shaw cell, which I'll explain kind of how that works in a minute. But I'm adding water into a glycerol solution, which is the glycerol is the high viscous and the water is the low viscous. So you can kind of see here as I add solution, the viscous fingers, as they spread through there. So yeah, that's the demonstration of it. You can definitely just pass that.
Is this just dyed water?
Yeah, it's just dyed water.
Give me your paper towels.
And so to kind of explain what that Healy Shaw cell I created, you have two acrylic plates, plexiglass acrylic plates, and in the middle of them is the high viscous fluid, which is glycerol in this case, and they have what is considered like a very large length compared to the space in between. And then you inject your low viscous fluid into the high viscous fluid, and that's what creates the Taylor instability, Softman Taylor instability. And so, looking at the Navier-Stokes equation, first we assume steady state, a very low Reynolds number, and that small, because of that small space in between the two cells, the two plates, we neglect gravity. And again, thinking about Reynolds number being inertial forces over viscous forces, because our viscous forces are dominating in this equation, in this situation, we kind of can also ignore our inertial forces, which leaves us with this equation over here, which is the change in pressure is equal to the viscosity times L squared of our velocity. And so then taking the integral of that twice, we get this equation here using boundary conditions and no slip. We can get a average of the velocity in the z. And we're also, I don't know if I mentioned this, but eliminating the x and y due to that. Like I said, we have a very large L compared to our z position. Yeah, no problem. So anyway, all of that leads to a control parameter that's based off of the capillary number. And with that, we are able to determine some information. That control parameter, as that control parameter increases, the width of the viscous fingers decrease. And so as a function of the capillary length, it is expected that the increase in viscosity ratio will result in decreasing width of those fingers. Other properties that can be shown are the increased viscoelastic fluid behind fingers with lower viscosity ratio, meaning the higher the viscosity, the longer the fingers will appear. And then the splitting between the tips, which I'll kind of show a little bit more, becomes more prominent with increasing viscosity ratio. So I've already kind of shown you the experimental setup, two acrylic plates. There are rubber bands that are about 1 millimeter that are used to space the two plates. So that B is about 1 millimeter. And then for my two liquids of higher viscosity, I use glycerol and corn syrup. Corn syrup is slightly more viscous than glycerol. And then dyed water is our low viscosity liquid. This is just a video, but since we've already seen it, I can kind of skip that.
Could you play? Why don't you show it? Because I think a lot of people did not see it.
Oh, sorry.
That's the corn oil, the transparent stuff.
So this is actually this one is the corn syrup. I did both glycerol and corn syrup in the demonstration was glycerol, and this demonstration was corn syrup. So yeah, I looked at glycerin with water, corn syrup with water, and then I also tried glycerin with air, but I had some struggles with that one. It didn't really work, mostly because of there was a lot of back pressure with glycerin and water, so I was able to be more consistent with the pressure and the speed at which I was pushing out the water. but with air, I could not control, like, be consistent with the velocity that the air was being pushed into the glycerol. So I did not use that for my analysis, and I just couldn't get it to work due to time constraints. But when comparing the glycerol, which has a viscosity ratio of 1,250 to the corn syrup, which has a viscosity ratio of 3,125, you can kind of see from here, you can notice that the higher viscosity has lower, on average, lower width between the fingers. The fingers are also longer in comparison. And then I counted on this surface here how many tips there were. And there were 16 tips in the lower viscosity situation with glycerol compared to 21 tips, so more tip splitting with the corn syrup. This is important and has important implications in oil extraction, where water is injected into the ground to push crude oil up into the production well. It's also important when you think about groundwater and soil contamination and how that contamination is going to spread. And then, interestingly for me, was the design of 3D biomaterials. There are different methods in which they utilize their understanding of this viscous fingering to create branched and tree-like structures that can be used to create leaf fenestrations, which you can see here, like so, as well as flow channels or producing neuron type structures. And then also, because of the season, dendritic crystal growth. So the creation of a snowflake kind of follows viscous fingering as well as growth of bacterial colonies. And this is just a video of, which is not going to even work. It's not that important, but it's a video showing the use of viscous fingering to create those flow channels in 3D biomaterial prints. So, it's really, again, it's showing the same thing, but there's multiple different ports. So when it comes together, it creates this branch-like structure.
Do you have the video online?
Yes, I can.
When you load it, can you show the demo live again, or does it take a long time to fill it up?
Let's see. Depends.
Well, I think let's applaud. If you want to see the live demo, getting some seating, gather around. If you can show it one more time.
Yeah.
But definitely before you leave, since this is all glycerol, I think you gotta go to the bathroom and
work
every little thing. It's not just water. It's a little sticky. I'm sorry we went over.
Do you have a minute?
Oh yeah. Would you? Yeah, great. Thank you. My hands are so sticky from the glycerol.
It's actually pretty good.
Something else I would decide on. Yeah, I'm currently
So like obviously for like groundwater.
Right.
And also it's like rehabilitation.
Yes.
That would be like 2 years.